so the University of Northampton is a really fun place to work. RNA is often the forgotten piece to the puzzle.
If there's one message that you want to give out to students who are like passionate about RNA, is there anything you want to put it out there? When people think about genetics, they think about DNA. But what if I told you major breakthroughs in medicine are not even about DNA? It's about how you read them. Hello there, everyone. I'm Adon. I'm a second-year biomedical science student studying here at the University of Northampton. And I'm joined here by Professor Karen Anthony, leading lecturer in RNA processing and therapeutics with over 20 years of experience. Hello there, Karen. Thank you for taking this opportunity and being here.
You're welcome.
Would you like to introduce yourself and explain it to someone without a science background, what you're doing and how you're doing it?
Sure, yeah. So I'm a professor of molecular medicine here at the University of Northampton. And my research interests span mostly RNA biology and how we can use RNA for therapeutic benefits. So I work on generally quite rare neurological diseases, mostly on Duchenne muscular dystrophy or some inherited forms of ataxia as well, and looking at how we can utilize knowledge of RNA for potential therapeutic benefits. So we're looking at RNA-based therapeutics or RNA-based gene therapies.
Right. So most people hear about DNA, but not about RNA. Why do you think that is, and why is RNA processing or RNA therapeutics something that's really important for our cell functioning, and why does it matter in like a disease perspective as well? What do you think so?
Yeah, so RNA is often the forgotten piece to the puzzle. So many people have heard of the central dogma. So DNA makes RNA, which makes protein.
Right.
And RNA is traditionally that bit in the middle people forget about. But without RNA, you wouldn't have the proteins to carry out the functions in the body. So after DNA is transcribed into RNA, the RNA molecules undergo a lot of processing within the cell in order to produce protein. And there's lots and lots of steps involved in that. And those steps are where things can go wrong in different diseases. And there are many, many genetic diseases where mishaps happen or mutations happen that affect the RNA processing, so at that RNA level. So it's the involvement of RNA in disease and the knowledge of and understanding that we've gained to say that mutations that affect RNA processing are a direct cause of disease. So that's where the interest comes from for me. And now also we've got some promising therapeutics on the way or already in the clinic targeting RNA.
How would you say the University of Northampton has shaped you as a researcher, and how has the environment kind of supported you of becoming who you are right now?
So the University of Northampton is a really fun place to work. So here we are a teaching-focused institution. So it's really great here that we're able to offer a lot of opportunities for students at all levels to get real access and involvement in our research. So we involve undergraduate students in projects, master's students through to MPhils, PhD students as well. And as a university, we've grown in our capabilities and facilities in life sciences, and particularly in the cell culture and molecular biosciences. So since I've been here, the capacity and amount of equipment we've got has vastly increased. And actually, the university have been very supportive in our pursuit of funding to acquire new equipment and develop facilities even further. So I'm really excited for where we're going with that, because we're about to launch an advanced imaging facility, for example, at the university, where we're able to offer access to more advanced techniques. At the University of Northampton, we're currently in the process of consolidating various pieces of advanced equipment, such as this scanning electron microscope, and packaging it into an advanced imaging facility that we're able to open up to external users. Okay, so we're currently in the laboratory that's been earmarked for refurbishment for our advanced imaging facility. So this time next year, this lab will look very different, and we'll have all sorts of advanced microscopy equipment in here, providing access for students and also researchers at the university.
So a lot of people hear about CRISPR, and like it's a widely known thing right now, and like how we can edit the gene and the DNA. But why should we focus on more RNA-based approach rather than the DNA? Why would you think so?
So I wouldn't say it's that we absolutely should focus on RNA. It's more about having a toolbox, right? So what we have now is a gene therapy toolbox, and there are lots of tools in that box. So sometimes it's best to target the DNA, sometimes it's best to maybe do a different approach, and it's about selecting the best tool for the job. So what CRISPR might be an excellent approach for one disease or for one particular group of patients, but it might not work for others. And so having a toolbox full of different tools enables you to develop different strategies for different diseases, different groups of patients. And with the RNA-based gene therapies, there's been a lot of work looking at developing therapies more on a precision medicine-based approach, so at specific, unique groups of patients.
Also, I think AI is something that we've already delved into the craziness of it. We know what AI can do nowadays. And do you think there's any way which in like AI or bioinformatics, for instance, or like there's ton of stuff now because of AI? And do you think it actually helped you in this RNA researching and like from analyzing your stuff, or like how has universities equipments kind of help you for that as well?
Yeah, so you can't avoid the onset of AI across any aspect of life now, and research is no different. So we are using AI-based tools in all sorts of areas across our research. So if we're thinking about drug design, for example, there are now pipelines and AI-based tools that enable us to better predict the effect a drug might have in cells. So traditionally, we would have to experimentally test lots of different candidates of drugs or molecules that we're interested in. But now what I'm finding is we're able to do a lot of that initial testing and candidate selection in an in silico environment. So using AI-based tools to better predict and enable us to choose the best candidates to test in cells. So it's saving time and money and reagents in the lab as well.
Looking ahead, what you think is your research heading to? Like you've done 20 years now, and that is indeed like it's not easy. And how would you just like focus on just researching on RNA? What made you go into RNA in the first place? Can you give a bit of history about that?
Yeah, so I guess my research journey started with my PhD, like it does for most people, and that project was looking at the involvement of RNA in neurodegenerative diseases. So that's where my interest came from, and I was doing very fundamental science. So there's a lot of hype and excitement about being translational and working on projects that have a very short impact timeline to patients, but actually a lot of work is still needed at the very fundamental and basic science level, which is where most of my work has been. So I started off, in that realm, working very much on basic science, trying to understand the basic principles of how RNA molecules might work in the cell and what might be going wrong in disease environments. And that led me to various different roles across my career, including working more on clinical work in Duchenne muscular dystrophy. So I've worked both on basic science as well as more preclinical science and clinical science. And I suppose that's the driver really, is seeing the impact increasing our knowledge of basic biology has on that sort of preclinical and then clinical development. So, and it's a fast-moving field. So, the RNA therapeutics field is moving very rapidly. We can go from an idea of a drug concept through to clinical development much more quickly now than previously. There's a bit more precedent in the field as well, and so that's a driver, is sort of knowing that. That what we are doing could potentially be translated into the clinic.
Also, if there's one message that you want to give out to students who are like passionate about RNA, who wants to come to the University of Northampton, is there anything you want to put it out there?
I think it's my advice is always just to be proactive and to take opportunities when you see them and when they come, and to do your research and to be proactive and put yourself out there. Don't be afraid. There's no stupid questions.
Exactly.
The worst that can happen is people can say no if you approach us. Yeah.
Well, thank you for this. This means a lot. Yeah. Yeah, that's about it.
All right.
All right.